Today's topic is nutrigenomics. Nutrigenomics, it's chapter 27. Those who have the nutrition science textbook, chapter 27, page number 478. If you have a different edition of the textbook, please mention the page number in the chat box. Nutrigenomics, page 478. And if anyone is joining late, please help them out with the topic name. 485 in the new edition textbooks. So nutrigenomics. So before understanding nutrigenetics or nutrigenomics, let's discuss on the basics of genetics. Okay, what is gene? What is genome? DNA, RNA. Okay, so here you can see on this black picture, which is on the right side, okay, we have a cell, and each of our cells, they have a nucleus. Inside the cell nuclei, you have your genome. Okay. A human genome has more than 30,000 combinations. Okay. So this is a human genome which you find inside a cell. Okay. So because of this, see, for example, if you find a piece of flesh, okay, if you find a piece of flesh or bones unearthed, okay, do you think it is possible to recognize whose flesh it is? Is it an animal's flesh, a human flesh? And even if it is human, is it a male flesh or a female flesh? Is it possible to identify? Yes, you can identify. Okay. And this is because how you can identify it is because the cells, it has genome. Okay. Animal genomes are separate from human genome. The combinations that humans have in their genome, okay, genome is a sequence of 30,000 and yet to be found. Okay, more genome sequences are to be found. So far we have around 30 to 35,000 genomes known. Okay, so human genome is different from any animal genome. Okay, so it is because of this, when you do analysis with a single cell, you can identify whose cell it is. Is it a human cell or an animal cell? And even if it is a human cell, is it a male cell or a female cell? That can also be identified. Okay, so inside a cell you have nucleus, inside the nucleus you have genome. Okay, chromosomes, different combinations of chromosomes come together and form a genome. And if this is a chromosome, okay, how many chromosomes does a human being have? How many pairs of chromosomes? Yes, we have 23 pairs of chromosome, with the 23rd pair being the sex chromosome. The 23rd pair of chromosome determines, are you a male or a female? Okay, male or a female. XY is male, XX is female. Okay, remaining 20 through 20 up till 22nd pair of the chromosome, males and females have the same structure. We all have same internal structures except for the reproductive organs, that is determined by the 23rd chromosome. Okay, so if you strip the chromosome, if you take one chromosome and you strip it down, you pull it out, okay, like a string, when you pull it out, that's how you get your DNA. This double ribbon, when this ribbon like structure is parallel, okay, you can see this helix kind of a structure. Okay, two ribbons are parallelly running. Two ribbons means it is DNA. Okay, if we take only one of this yellow ribbon, okay, that is RNA. Okay, two RNA ribbons come together and they form a DNA. Okay, so DNA also has a sequence. from point A to point B. Okay, that would be one DNA sequence. And within the DNA sequence, you will have the genes. Okay, two to three sequence of two to three sequences will form a particular kind of gene. Five to six sequences will form another kind of gene. Okay, so this is how the basics of genetic is, from cell till the DNA and till the genes. Okay, within the cell you have genome, um, chromosomes. Chromosomes form the human genome. You take one chromosome. Okay, we have 23 pairs of chromosome. You take one chromosome, you strip it down, you will get the entire DNA structure. Two ribbons. Okay, if you talk about only one ribbon, that is your RNA. Okay, and these blue orange markers that you see, these are sugar and protein molecules. Okay, sugar and protein molecules, different types of sugar and protein molecules. Okay, so these also have a specific sequence and they form a gene. Okay, is it clear to all? The basic of genetics, especially for those who are not from a science background. So if you see the difference between gene and a genome, okay, the functional piece of a DNA is a gene. As I mentioned, lot many genes in different permutations and combinations, they come together and they form a DNA. Okay, my DNA will be different from yours. Why? Because the genes that are in my DNA in a specific sequence is different from your DNA. Okay, but genome, do you think my genome and your genome would be same? When we talk about genome, okay, yes, genomes would be same. Okay, but DNA, because of certain sequences, DNA more or less is majority of our DNA, human DNA, to an extent is basic, same. Okay, it depends on ethnicity. We all belong to the same ethnicity, so we have that similarities. But with certain genes, okay, in a specific sequence, that determines from which part of the country we may be. Okay, what we have inherited from our family. Okay, this is all defined by the sequences of your genes. Okay, and gene is made up of few 100,000 base pairs. You can see here the base pairs, which is this yellow, blue, blue, orange. Okay, these are the pairs ATCG. Okay, so these are the pairs. Okay, few thousand base pairs will form one gene. Okay, and it is non-repetitive. Genes should not be repetitive. If the genes are repeating, it will lead to some form of genetic disorders. Okay, and genes, you will find genes on the chromosome. You can see here chromosomes are the ones that carry the genes. Okay, and when you study about the genes, different sequence of genes, you are studying about genetics. Okay, when it comes to genome, okay, all the haploid set of the DNA is a genome. Genome is found inside the cell. Okay, there are so far what we know is around 6.4 billion bases in the genome. It can be much more than that, so far what we have found. Okay, it is also non-repetitive, and it is found in the nucleus of the cell. And when we study about genome, we call it genomics. Okay, the study of genomics is very vast because you're collecting data about the human existence. Okay, so it is a very vast study. Even the evolution of humans. Okay, when we want to study about the evolution of humans, we study about genomics. Okay, and this is the difference between DNA and RNA, as I mentioned. When we have the double ribbon structure, a double helix structure, this is DNA. Okay, when we talk about only one single ribbon, okay, two ribbons have to come together to form your DNA. When we talk about one single ribbon, it is RNA. Okay, so these are the base pairs. cytosine, guanine, adenine, thymine. Okay, these are the base pairs. These four sugar molecules, okay, sugar and protein molecules, they interact with each other, okay, in such a way they form so many permutations and combinations on your DNA that gives you your physical features. Okay, how everyone is different from each other. It is because how these base pairs, okay, are different, are combined differently in your genes. Okay, is it clear to all so far, the basics of your genetics? And you can see here all these are nitrogen based component. Okay, is it clear? Is the hexagon structure, is it clear to, is it visible to all on screen? Can you see the nitrogen molecules here? On each of these components, on thymine, adenine, guanine, okay, their carbon structure is given, okay, organic structure is given. Each of these molecules, they have nitrogen component. So which macronutrient is important for the health of your DNA? Which macronutrient is important? Yes, amino acids, which comes from protein. Okay, you get nitrogen, you get this N. Okay, N is nitrogen. You get the nitrogen only from your protein. Okay, protein is the only macronutrient that can provide you nitrogen. Okay, so this is the importance of having protein, correct, adequate amount of protein in your diet. It uh, it makes sure at your DNA level, at your genetic level, from that level you are healthy. Okay, so what are haploid and diploid cells? Haploid cells are just one set of chromosomes, as we have mentioned. We have 23 pairs of chromosome, 23 pair. Okay, so when we talk about only when we divide it, okay, you take a scissor and cut it in between. Okay, so you just get the haploid structures. And when they are in pairs, this is how they are in pairs. Okay, so when we have them in pairs, it is diploid. Okay, so just imagine if you have one more string attached here, like a triplet, not a pair. Okay, if you have one more string attached here, okay, if anyone is born with one more string attached, do you classify that as a genetic disorder? Yes, it would be. For example, Down syndrome. Down syndrome. Down syndrome's other name is trisomy 21. Trisomy 21. Tri means three. Okay. Somi is chromosome. Chromosome. Okay. So somi. What is 21? On the 21st chromosome, instead of it being just diploid, it has one more additional chromosome attached to it. So it has three chromosomes attached on the 21st chromosome. When you sequence it on the 21st chromosome, when there is one additional pair, okay, when there is one additional string, making it three, not two, that is trisomy 21. That's how you get Down syndrome. Okay, now coming to the difference between nutrigenetics and nutrigenomics. Okay, nutrigenetics, your genes affect how your body responds to food. Okay, genes of it's a classic example, I have given it in previous classes as well. Okay, let's take South Indians and North Indians. Okay, do they tolerate milk equally, the way how they respond to milk or milk based products? Is it going to be the same? No. Okay, North Indians can tolerate milk or milk dairy based products, for a longer period of time. Okay, even after their 50s and 60s, they will not have any issue with milk. But South Indians, it's not the same case. Why? Genetics. Okay, their genetic is affecting how their body responds to food. Okay, their genetics is affecting how the body responds to food. That is nutrigenetics. Okay, next, nutrigenomics. Nutrigenomics. What you eat affects your genes. What you eat affects how your genes behave, or what you eat affects your genes. For example, if I am having a diet that is high in fertilizers and pesticides, okay, I am not washing my fruits and vegetables properly, okay, I'm having mostly raw or uncooked food and all, okay, so will that impact the diseases that I may get in the future? Will that impact? It will definitely. Okay, the risk of getting cancer, my risk of getting cancer, my risk of getting any form of deformity, okay, or even to my kids, okay, even to the future generation, okay. So what is happening here? Nutrition, good or bad, it can cause some genetic level variation. Your a nutrient can cause genetic mutation. What you eat can cause a DNA level mutation. Okay, it can change how your genes behave. It can either cure your certain diseases or it can give you some new diseases. That is nutrigenomics. Okay, genes will affect how your body responds to food, and uh, food can also affect how your genes would behave. So this is nutrigenetics and nutrigenomics. Is the difference clear to all? Okay. Now coming to the principles of nutrigenomics, that is this chapter, principles of nutrigenomics. Why we call it as principles? These statements that are given on page number 479. Okay, those who have the textbooks with them, there is a topic, principles of nutrigenomics. Only in short form I have mentioned on the slide. The entire statements are mentioned on the textbook. If you have the textbook, you can refer the textbook. We will discuss each of the statements. Why it is called principles of nutrigenomics? Time and again, these statements have been proven to be right. Okay, time and again, these statements are evidence based and they are proven to be right. Okay, large number of studies have proven this assumptions as true. This is the reason why they became principles. These are the guiding 5 statements in nutrigenomics. Okay, so the first statement, gene expression can occur directly or indirectly, which means your gene expression or DNA structure can be modified by the action of nutrients of and bioactive components of the food that human beings tend to take. Okay, and this modification can happen directly or indirectly. For example, let's say we are having some fertilizer or pesticide laden food, not correct, not we are using harmful uh, pesticides which are actually banned, which should not be used in agriculture. Let's say some part of the country is still using it, okay, is still using such banned pesticides in their agriculture. Okay, so these pesticides, through the food, it can change the human genome. It can modify human genome either directly or indirectly. Directly, the pesticide itself is responsible to cause some mutation. The pesticide itself is responsible to cause some mutation. Indirectly, the pesticide is causing some inflammation or certain disease conditions in your body. which became chronic, you didn't treat it, and this chronic inflammation or chronic disease condition changed something in your DNA. These chronic diseases can also cause mutation in the body. Okay, chronic diseases can also change your DNA. Okay, so either directly or indirectly, a pesticide or any biological food component can cause modification in your genes. Is it clear? The first statement, is it clear to all? The second statement is about your diet. Diet is a risk factor for certain diseases in some individual and also under certain circumstances. For example, let's say, long-term medication can also change DNA. Long-term medication, especially when it comes to steroids and all, okay, can cause a DNA level mutation. Okay, next is diet is a risk factor. Let's say, for example, both my parents are diabetic. Okay, let's say both my parents are diabetic. So, am I supposed to take care of glucose in my diet? Diabetes has not started in me yet. Okay, but if you take my family history, let's say, for example, both my parents are diabetic. So, me as an individual who belongs to the same generation, okay, who belongs to the generation who had diabetes, okay, should my diet influence my choices? It will, okay, it will, because I am at a risk. I am at a risk. Family history is a main category of predicting what kind of diseases you will have in your future. Okay, we as individuals, we may not have any idea what diseases is hidden in our genes. Look at your grandparents, look at your own parents. What all diseases are they undergoing? You are at the risk of getting the same diseases. Okay, so diet is a risk factor. And usually when we talk why diet is a risk factor, will my diet be completely different from my parents' diet? What do you think? Will my diet be completely different from my parents' diet? In a general case, in a general scenario? No, it will not be. Okay, because growing up till certain age, okay, early adulthood or mid adulthood, parents as well as me, we are having the same food. We are having the same staple food. The same food that gave them diabetes or made them ill, the same food which I'm continuing right now also has the risk potential to put me into diabetes as well. Okay, so diet is a risk factor. Okay, whatever your parents have been carrying at what you will get in the future based on the diet. If diet is the same factor, okay, diet is the same factor for your parents and you, okay, diet becomes the risk factor of certain diseases as well. Okay, next is chronic disease onset, incidence, progression, or even severity is directly influenced by diet regulated genes and their common variants. For example, if I am having antioxidant rich diet, okay, a chronic disease has just started. For example, I am suffering from asthma since teenage. Okay, let's say, for example, since teenage, I am suffering from asthma. Okay, and if I am focusing on more antioxidants in my diet, will the triggers of asthma reduce in me, or will the triggers of asthma increase? It will reduce. Okay, even if it is asthma is a chronic disease. Okay, it's not usually children may get over asthma uh, by teenager, but if you get asthma after certain age, okay, in late teenage or after teenage, if you are diagnosed with asthma, it usually stays with you for the lifelong. Okay, so in such situations, if we focus on the diet that are not inflammatory, if you focus on the diet that is antioxidant rich, definitely, even if the chronic disease has begun in your life, the way how it will progress or the way how it is going to get severe, it completely depends on your diet. Okay, I know that I am asthmatic, but but I'm keeping myself out always in the dusty environment. I'm always having cold food. Okay, even if I'm having slight cough or sputum, I had an allergic reaction, I had lot of mucus production, I'm not eating anything hot, I'm only having cold food, not having antioxidant rich food. Okay, it will worsen the situation. It will worsen. Diet can worsen your chronic disease. Okay, but if I'm being more careful, antioxidant rich food is not the treatment for asthma, not at all. Okay, but your diet can slow down. Okay, slow down the severity of your chronic diseases. Okay, all fruits and vegetables. That is the example of antioxidant. All fruits and vegetables, vitamin C, vitamin E, omega 3. Okay, all fruits and vegetables, as much as you can have, that is your antioxidants. And a handful of nuts. That's it. Seasonal fruits and vegetables. So, next principle is your genetic makeup. So, the influence of diet on health and disease is likely to be reflected by genetic makeup. Okay, for example, uh, two children who have been affected by cholera, two children from completely different family. Okay, let's say one child is from a BPL family, below poverty line family. Okay, and one child is from a middle class family. Two children of the same age got cholera. The way how the child, in a general scenario, okay, not exceptional cases, in a general scenario, the way how these two kids will recover from cholera, is it going to be exact same or different? Which child may have to suffer a little bit more, or which child's recovery will take longer time? The child who comes from a BPL category, because there are high chances that the child who is from a BPL category comes from parents who, or genetic lineage, who had been malnourished. Okay, generational malnourishment is running in their family, and because of that, the risk of them developing diseases is higher. Okay, the recovery from diseases will be longer. It is completely on the child's genetic makeup and whatever current nutritional status the child may be going through. Okay, in a general scenario, the children who come from rural families or poverty, below poverty line families, they may take a slightly longer time to recover. Okay, from any infective diseases, because malnourishment, there would be underlying malnourishment, either hidden malnourishment or overt malnourishment will be present, which will affect the way how the child recovers. Okay, so that is genetic makeup. Your genetic makeup also decides how will you recover. Okay, how will you stay healthy, or how will you get affected or recover from diseases. Next, last principle is your nutritional intervention. So nutritional intervention, this is where the clinical counselors or genetic counselors role is intensified. Okay, so when you give nutritional intervention to any of your clients, you have to keep in mind what is the individual's current nutritional status. Are they malnourished? Are they deficient in certain areas? Okay, what kind of diseases or health risk do they carry from their family history? Okay, what are the nutritional requirements that you can tailor for this particular client, so that in future they will not have to face the same situation what their parents or grandparents may have faced. So this is nutritional intervention. Okay, based when you are dealing with one client, you have to go in depth about their family history, their current nutritional status. Okay, and based on their current nutritional status, if they are headed towards what diseases their family was carrying, okay, what nutritional changes you can do as a dietitian, that you can revert them from going to the same path. Okay, so these are the 5 principles of nutrigenomics. Is it clear to all? Next, coming to interactions. Okay, so genes or nutrient, how they interact with each other. So what is direct interaction? Okay, nutrients can directly cause some changes in your DNA. Nutrients can directly interact with the receptors that you have in the DNA and cause some changes. You don't require any other enzymes or any other proteins to come into play and cause some change, or any other factors in your body to come into play and cause some change. Nutrient themselves can directly go and attach themselves to the DNA and cause some changes. That is direct interaction. So you can see here an example of a direct interaction. So let's say this activator, this activator is a nutrient, okay, and this activator has a site in DNA where it can go and bind and cause some changes. Okay, the activator will not go and attach itself to anywhere on the DNA. Okay, the strand of the DNA has specific receptors. Strand of the DNA has specific receptors where activator can attach, latch on, and then a change will come into play. Okay, so there are specific sequences. You can see here ATC, AATG. Okay, if you remember the binding proteins we have discussed initially. Okay, this is a specific sequence in which, and when this sequence comes on the DNA, activator can go and attach here. It will not attach to any other sequences. It requires this specific sequence to get attached, for example. Okay, and when this attachment takes place, a change will start. Okay, and this change will go and affect the target gene. Okay, it could be the gene of your digestion, it could be the gene of your metabolism, it could be the gene of certain skin rashes. Okay, any sign and symptom that will come into play. Okay, it could be anything. So activator will require a specific binding site on the DNA and it will cause some changes. This is direct interaction. Okay, next is you have repressor. What repressor does? Let's say example, repressor is your antioxidant. Antioxidants are repressors. So repressors also have a binding site. Okay, when antioxidants will go to their specific binding site on the DNA and stay attached there, how much ever activators come to cause changes, this activator will not be successful in causing some mutation, because the repressor is already present there. The repressor is already present on some part of the DNA sequence, and because of the presence of this repressor, which could be an antioxidant or any anti-inflammatory agent, because this is already there, it came there first. Okay, a repressor latched onto the DNA first, and activators, how much ever activators come and latch onto their sequence, they are not successful to cause some mutation. They are not successful to cause some change in your healthy gene. So this is the function of good nutrients. Is it clear? Next is epigenetics. Epigenetics is everything that is around you. You look at you look at your surroundings, okay, and how your surroundings affect you at your DNA level. That is epigenetics. Okay, for example, let's say you're not born yet. Okay, you are talking about a fetus. Okay, fetus who is inside a mother's womb. Will the fetus have an epigenetics? Will the fetus have an epigenetics? Yes, the fetus will also have an epigenetics. Mother's environment, mother's surrounding, amniotic sac, okay, health of the uterus, health of the mother from within, mother's internal body, okay, that becomes the child's epigenetics, right? And when the child is born, just like all of us, we are living out, okay, everything around us which you are in direct contact, the air you breathe, the water you wash yourself in, the food that you eat, okay, the area where you live, okay, is it a rural area, is it a urban city, okay, all these things directly affects your genetics. Okay, the outside environment directly affects your genetics, and that is responsible for many of the diseases that you may carry: heart diseases, diabetes, cancer, metabolic syndrome, any rare diseases that you may get in the future. Okay, epigenetics have some or the other, it is related to these diseases some or the other way. Okay, so that is epigenetics. Next, coming to the tools in nutrigenomics. So there are four main tools that we have. Okay, genotype and phenotype. Genotype is not that everyone can see. It's in your genes, sequence of your genes in the DNA. That is genotype. Okay, phenotype is what physical features we can see outside. For example, blue eyes, brown eyes, curly hair, straight hair, fair skin, dark skin, okay, height, weight, okay. These things are phenotypes. But to have a curly hair, there is you will have a specific gene sequence. To have a straight hair or wavy hair, you have a different genotype. Okay, blue eyes have a separate genotype, brown eyes have a separate genotype. Phenotype is what we are seeing. We can see this person has a blue eye, or that person has a blue brown eyes, curly hair, straight hair. Is this person fair or dark? Okay, these are the phenotypes, characteristics, physical characteristics that we can see. Genotype is the sequence to get that. Okay, and then epigenetics, epigenomics, just now we have discussed. That is one way of identifying what could go wrong in your genetics, or what mutations can cause. Okay, genomics, study of the genome, okay, that we have studied, and it stores information in the cell to predict what can happen. Okay, a human genome, animal genome, they all can predict what can happen with this person. Okay, next we have proteomics. Proteomics, there are more than 1 million protein in your DNA sequences, and you study that. Okay, the way how these proteins and nucleotides are sequenced. Okay, what change in the sequence, or what change in the protein could cause some mutation? When you study all this, that's what we call as proteomics. Metabolomics, the study of molecules involved in cellular metabolism that would eventually depict the phenotype of an organism. Anything in any biological reactions that takes place in your body has a byproduct. has a metabolic. Okay, and these metabolites can also go and cause some genetic level variation. Metabolites that circulate in your system. Okay, metabolites that are found in your blood circulation, lymphatic circulation. Okay, from where do you get the metabolites? When proteins are broken down, when the carbohydrates are broken down, fats are broken down. Okay, when you utilize vitamins or minerals for specific functions in your body, something out of that reaction, there is some byproducts. Okay, these byproducts are metabolites. Metabolites also tend to cause some genetic variations. Okay, so when you study about this, which metabolite is causing what kind of genetic variation, okay, that's what we call as metabolomics. Then transcriptomics, the study of RNA or any transcript enzymes, okay, that would depict what actually happens in the cell. Because if you remember the previous slide, if you remember the previous slide, this activator, once it got attached to a sequence in the DNA, transcription begins. Okay, transcription means a change. A change has begun. Okay, for this change to start, you will require certain enzymes called transcriptase. Okay, so when you study about these changes, this transcription, and when you study about the enzymes called transcriptase enzymes, which are actually causing the change, okay, the activator has just attached itself to the DNA. But what happened after this attachment, that is also a study. That also scientist has to decode. Okay, after this attachment, what actually got activated? How did the change happen in the cell? Okay, that is also a study. When you study that, that is called transcriptomics. Okay, this is what you have to know. These are the terminologies that you have to know from this chapter. Okay, in detail they have mentioned about genomics, proteomics till page number 484. That you don't have to read. Okay, just come to this part of the recording where I have explained the name, meaning of each of these terminologies. That's enough. That's enough for the examination. Is it clear to all? Next, coming to the bioactive components of food. Bioactive components, when we talk about bioactive components, usually they are your vitamins, minerals, antioxidants, anti-inflammatory agents, okay, anti-carcinogenic agents, which have certain effects apart from giving you nutrition. Okay, apart from giving you nutrition, what other functions they can do in your body to safeguard your health? Okay, so these are bioactive components. No need to go about the target and action. Okay, no need to go in depth at all. Just know the meaning of the medical terminologies. That's it. So bioactive components of food, it it will lead to cell growth. Okay, your nutrients, your vitamins, minerals, antioxidants, they allow your cell to grow in a controlled manner. When cells grow in an uncontrolled manner, what do you get? When cells grow in an uncontrolled manner, what do you get? You get cancer. Okay, so they prevent carcinogenesis. Bioactive components of food, they prevent carcinogenesis. Carcinogenesis means formation of cancer. Okay, you can see here the pink cells and the blue cells. Okay, pink cells have a controlled cell differentiation and cell growth. Okay, it is very much regulated. But this blue patch, the blue tissue here, okay, uncontrolled cell growth, not in a specific line, not in a specific order, not even the healthy size of the cells. Okay, so this is a tumor, this is a cancer. Okay, so to prevent that, also you require bioactive components in food. It helps in healthy cell differentiation. Cell differentiation is what keeps you young. Okay, cell differentiation is what keeps you young. Okay, as you age, your cell differentiation slows down. When you exercise, okay, when your body is able to get rid of the toxins, cell differentiation pace is high. Okay, you cannot change your DNA. You can only work according to the risk factors you already carry. Okay, changes in the DNA are not under human control. You can only work with the information that you have in your hand and choose your diet accordingly. Bioactive components of food will also lead to hormone regulation, and hormone regulation is important in metabolism. Okay, irregular hormones can also trigger cancer. It will reduce the inflammatory response. It will reduce the inflammation, unwanted inflammation. It can also repair damaged DNA, like antioxidants and anti-inflammatory agents. They can repair your damaged DNA. Damaged DNA can lead to certain mutation and disease conditions. It can lead to apoptosis. Apoptosis is cell death. Okay, cell killing itself. Usually apoptosis happen when one cell gets infected. It understands that a virus or a bacteria has come inside. The cell will kill itself so that the bacteria or the virus does not go out and does not cause any more infection. Okay, or any toxic agent has entered the cell, or if the cell has turned cancerous, the cell will kill itself. This is your own biological clock. Okay, your body has the system to identify a cancerous growth and kill itself. Okay, so that it does not spread, it does not differentiate. So that is apoptosis. Bioactive components of food will also lead to apoptosis, giving the cell the capacity to identify that something is wrong with itself, and let's just end it so that whatever carcinogenic agent is there or whatever infection is there, it should not jump from one cell to another. Okay, so these are the functions of bioactive components of food. Next is chronic disease and nutritional genomics. You just have to focus on the main arrow that is going downward in the center. Okay, that should be your main focus here. How a disease condition, how diet first would lead to disease condition, and how disease leads to some changes in the diet. That is secondary, but primary is how diet could lead to certain disease conditions. Okay, so diet, that is the most earliest marker of the food bioactivity. Okay, nutrients can have cellular level changes. Nutrients can cause cellular level changes. They are the earliest markers of changes in your DNA, your diet. Okay, so pivotal changes in gene expression can take place. If your diet is unhealthy, pivotal changes means which cannot be reversed back that easily. Okay, once you are diabetic, okay, or once you are in a pre-diabetic stage, okay, and once you are diabetic, you have two stages: pre-diabetic stage and diabetic stage. Which do you think is easy to reverse. Go back to normal. Yes, it is the pre-diabetic stage, which you can go back to normal. Diabetic stage, it's very rare. It's very, very rare to switch off the diabetic stage in your body. Okay. So when you enter into diabetes from pre-diabetic, when you enter into the diabetes, pivotal changes has taken place in your gene expression. Okay. Usually unreversible changes have taken in your gene expression. Now you have an abnormal gene expression. One that is, for example, in diabetes, what is the abnormal gene expression? What abnormality is happening in diabetes? Let's say it's about type 2 diabetes. Pancreas are no longer producing insulin. Pancreas are no longer producing insulin. That's an abnormal gene expression. What could have been a normal gene expression? Pancreas are producing enough amount of insulin, okay, and your cells are responding to it. What is abnormal gene expression? Pancreas are not producing insulin. Pancreas have stopped producing insulin. That is an abnormal gene expression. What will happen? The cell will show dysfunction. Okay, your cells will not have enough energy. The beta cells that were supposed to produce insulin, they are not producing insulin, and they will atrophy. The cells will get smaller and smaller in size, and they will stop functioning. Okay, the cells that are supposed to produce certain hormone, when they stop producing that hormone, that is no longer to be of a specific size. Okay, they reduce in their size, they reduce in the number as well. Okay, and the organ will undergo atrophy. It will decrease in the size. Okay, so cellular dysfunction will take place, and then you are in the disease category. You are diabetic. Okay, once you are diabetic, it will again lead to a lot of cellular dysfunction. It can lead to retinopathy. It can lead to, if it is uncontrolled, okay, uncontrolled diabetes can lead to further cellular dysfunction. Nobody gets, if you're suffering from insulin resistance or if you are in a pre-diabetic stage, will you get retinopathy or neuropathy? Will you get any complications of diabetes? You are in an insulin resistant stage or you are in a pre-diabetic stage. Will that lead to retinopathy or neuropathy? It will not, because retinopathy, neuropathy, they are chronic conditions. It takes a lot of time. It takes years to show up. Okay. So disease is diabetes. Okay. And because of diabetes, a person is suffering from diabetes from the past 10 years, 20 years. Slowly, even if they're managing it, okay, but slowly diabetes, which means high blood sugar levels, and years of high levels of blood sugar levels will lead to cellular dysfunction at various sites in your body. In your retina, it is retinopathy. In your nerve cells, it is neuropathy. In your kidney, it will be nephropathy. Okay. In your heart, it will be cardiac disorders. So at various sites in your body, 20 years of diabetes, 20 years into diabetes, okay, high levels of blood sugar levels most of the time. Inflammation has started here, there, everywhere. Okay, now the disease is causing further cellular dysfunction. And when these cellular dysfunctions start at different parts of your body, again, in different organs, abnormal gene expression will take place. Retina, which is supposed to show you clear images, now it will stop showing you clear images. You will not be able to see. You may turn blind. Okay, and when you turn blind, when the retinopathy has set in, can you go back? Or when the neuropathy has set in, can we go back? Is it reversible? No, it is not. Pivotal changes in gene expression has taken place. Okay, now you cannot go back. Is it clear how chronic diseases and nutritional genomics work? Is it clear to all? another a risk of this is how your family life, okay, your family history impacts your risk. People were having doubt, somebody had shown a doubt, can you change your DNA? Okay, so let's say these are the low genetic risk people. Okay, the on the left side, low genetic risk people, people who do not have a family history of cardiovascular diseases in their family. Nobody, neither the grandparents nor the parents, nobody was suffering from any heart related diseases. Okay, when they have a healthy lifestyle, their vein, their arteries will be clean. Their arteries will not have any plaque. Okay, any fat cell deposits with a healthy lifestyle. But even though they do not come from a family of cardiovascular diseases, but if they choose to have a very bad lifestyle, even though they nobody had it in their family, they can start the disease. Okay, these people, they can start the disease. Okay, fat cells can start depositing in their blood vessels, even though the family history was safe, but the lifestyle made it unsafe. Okay, now coming to the high genetic risk of cardiovascular diseases from both the sides of the family, maternal and paternal. From both the sides of the family, cardiovascular diseases are present. Even if they, and in this generation, even if they follow a healthy lifestyle, to a certain extent, they will always have the risk of developing cardiovascular diseases, even with a healthy lifestyle. Okay, they can't run away from it. They have to be very cautious about their lifestyle, the food that they have. Okay, and with poor lifestyle, their risk is very high. Okay, even 10 to 20 years earlier as compared to their previous generation, they may get the disease. Okay. So that's about Nutrigenomics. If you have any queries, you can mention